Everyone, welcome back to my channel. Don't forget to click subscribe if you want more daily updates on Microbe Mania, where we are going to study about interview questions and answers of microbiology in pharmaceutical industry.
Welcome to today's video. We'll be diving into the top instruments found in a microbiology lab. These tools are essential for everything from studying bacteria and fungi to diagnosing infections. Let's explore what each instrument does and why it's so important in microbiology. Let's start with one of the most essential tools in any microbiology lab, the microscope. Microscopes are absolutely crucial for microbiologists because they allow us to see the invisible. They let us zoom in on bacteria, viruses, fungi, and other pathogens, helping us understand their structure, behavior, and how they interact with the world around them. Without microscopes, studying these tiny organisms would be nearly impossible. There are several types of microscopes, each serving a unique purpose in the lab. Let's break them down. Compound microscope. This is the most common type for studying bacteria. It uses multiple lenses to magnify specimens, offering a magnification power of up to 1000x. It's perfect for looking at things like gram-stained bacteria, where you need high magnification to see cell shapes and arrangements. Stereo microscope. This type is great for larger samples that don't require super high magnification. It provides a 3D view, which is ideal for observing larger organisms or objects like fungal colonies or insect specimens at lower magnifications. Fluorescence microscope. Fluorescence microscopes are used to identify specific components within a sample. These microscopes use fluorescent dyes that glow when exposed to UV light. This is incredibly useful for pathogen detection, as you can highlight specific microbes or structures for easy identification. Electron microscope M. When you need extreme magnification, electron microscopes are your go-to. They include transmission electron microscopes TEM, which allow you to look at the internal structure of cells, and scanning electron microscopes SEM, which give detailed 3D images of the surface. These are used to study things like viral particles or intracellular components at atomic scales. Phase contrast microscope. Unlike traditional microscopes, phase contrast microscopes don't require staining. This is great for observing living cells, as it enhances contrast without killing the sample. It's perfect for studying motility, or how cells change shape and move. Some of the leading brands in microscopy are Olympus, Leica, and Nikon. These brands are well known for their quality optics and reliability in microbiological research. Microscopes are used for a variety of tasks in microbiology. Here's how they help in everyday lab work. Bacterial identification. By observing cell morphology, like shapes, arrangements, and reaction to stains like gram stain, scientists can identify bacterial species quickly. Pathogen detection. For identifying pathogens, especially in clinical samples, fluorescent dyes are used with a fluorescence microscope to make the pathogens glow, allowing for rapid diagnosis. Cell behavior observation. Microscopes also allow us to study live cells in action. We can observe their movement, replication, and how they interact with other cells or substances in their environment. Next up, we have another essential piece of equipment in the microbiology lab, the incubator. Incubators are vital for cultivating microbial cultures by providing a controlled environment with stable temperature, humidity, and sometimes even CO levels. Without incubators, growing and studying bacteria, yeast, or mammalian cells would be nearly impossible. There are different types of incubators, each serving a specific purpose, depending on the needs of the organism you're growing. Let's take a look at the most common types. Standard incubator. The standard incubator is the most widely used type, especially for growing bacteria. It maintains a constant temperature, typically around 37 degrees Celsius, which is perfect for culturing bacteria that thrive at body temperature. It creates a stable environment where microbial growth can occur at an optimal pace. CO incubator. For mammalian or plant cells, the CO incubator is the go-to tool. It regulates CO levels, ensuring that cells grow in conditions similar to those in the human body or plant tissues. This is especially crucial for growing cells that rely on specific CO concentrations for proper metabolic functions. Shaker incubator. A shaker incubator combines two key features. It provides the right temperature for growth, but also shakes the cultures. This makes it perfect for growing liquid cultures, like bacterial or yeast suspensions, ensuring that the microbes stay well mixed and have even access to nutrients. Anaerobic incubator. An anaerobic incubator creates an oxygen-free environment, allowing you to grow anaerobic bacteria, which can only survive without oxygen. This type of incubator is essential for studying organisms that thrive in extreme conditions, like certain types of bacteria that cause infections. Some of the top brands that manufacture incubators include Thermo Fisher Scientific, Eppendorf, and BMT. These brands are known for their high quality and reliable equipment, trusted by labs all over the world. Incubators are used for a variety of tasks in microbiology labs. Here's how they're put to work in everyday research. Microbial culture growth. Incubators provide the perfect environment for growing bacteria, fungi, and other microorganisms under optimal conditions. Whether it's bacterial colonies or yeast cultures, incubators help ensure consistent growth. Cell culturing. For research involving mammalian or plant cells, incubators maintain a controlled environment to keep these cell lines alive and growing, which is crucial for everything from cancer research to genetic studies. Temperature-sensitive studies. Many organisms have strict temperature requirements for growth. Incubators ensure that temperature-sensitive organisms can grow under consistent conditions, giving researchers the best chance for accurate results. Whether you're growing bacteria, culturing mammalian cells, or studying anaerobic organisms, the incubator is indispensable in any microbiology lab. It helps create the perfect conditions for microbial growth and ensures that experiments are consistent and reliable. Let's dive into one of the most important tools for ensuring a sterile environment in the lab, the autoclave. Autoclaves use high-pressure steam to sterilize equipment and materials. By killing harmful microorganisms, they ensure that everything from tools and growth media to waste materials are free of contamination. Autoclaves are a must-have for any microbiology lab to maintain a clean and safe workspace. There are different types of autoclaves, each designed for specific sterilization needs. Let's go over them quickly. Gravity displacement autoclave. This is the most basic autoclave design. Steam enters the chamber, displacing the air, which allows the steam to sterilize materials. It's perfect for sterilizing simple items like glassware and tools. Pre-vacuum autoclave. The pre-vacuum autoclave takes it up a notch by removing air before introducing steam. This allows the steam to thoroughly penetrate complex materials like wrapped instruments and porous objects, ensuring they're sterilized completely. Steam autoclave. The steam autoclave is the basic version that uses steam to sterilize equipment. It's often used for sterilizing glassware and media before experiments. Some of the top brands for autoclaves include Tutnauer, Sterigenics, and Belimed. These brands are well regarded for producing high-quality, reliable equipment that microbiologists around the world trust for sterilization tasks. Now, let's talk about how autoclaves are used in everyday microbiology. Here are some key use cases where autoclaves are essential. Sterilizing glassware. Before any experiment, it's essential to sterilize tools like Petri dishes, test tubes, and other glassware to avoid cross-contamination. The autoclave ensures that all these tools are free from unwanted microorganisms before use. Media sterilization. In microbiology, we work with agar plates and broths to grow microorganisms. It's crucial to sterilize these media to kill any existing bacteria or fungi. Autoclaving the media ensures that only the microbes we introduce are allowed to grow. Waste disposal. Autoclaves also play an important role in disposing of contaminated materials safely. After an experiment, items like used Petri dishes, swabs, and gloves can carry harmful bacteria or viruses. The autoclave sterilizes these materials before they are safely disposed of, preventing the spread of infections. Autoclaves are indispensable in microbiology labs for ensuring sterilization and preventing contamination. Whether it's sterilizing tools, media, or disposing of waste, autoclaves play a critical role in keeping labs safe and experiments accurate. Now, let's talk about one of the most essential tools in microbiology, the Petri dish. These shallow, flat containers are used to grow microorganisms on a solidified nutrient medium, often agar. Petri dishes are vital for culturing, isolating, and studying individual colonies of bacteria, fungi, and yeast. Whether you're working in a research lab or clinical setting, Petri dishes are a staple of microbiological work. There are a few different types of Petri dishes, each with specific purposes. Let's break them down. Standard Petri dishes. These are the classic, round dishes made from clear plastic or glass. They're used for growing a wide range of microorganisms, from bacteria and fungi to yeast. They're perfect for general culturing needs. Sterile Petri dishes. Sterile Petri dishes are pre-sterilized to avoid contamination from environmental microbes. These are especially important in aseptic techniques, where avoiding contamination is key, particularly in clinical settings or when handling sensitive samples. Multi-well plates. Multi-well plates are essentially Petri dishes divided into smaller compartments or wells. These are used for high-throughput testing and are commonly found in diagnostic microbiology. They allow researchers to test antimicrobial susceptibility or perform screening tests on multiple samples at once. Agar plates. Agar plates are Petri dishes that come pre-filled with agar medium, a gel-like substance that provides nutrients for microbial growth. These plates are ideal for isolating individual bacterial colonies and performing tests like antibiotic resistance assays. When it comes to reliable Bunsen burners, top brands like Fisher Scientific and Cole-Parmer are trusted for their easy-to-control and adjustable burners. These brands provide high-quality burners that are essential in any lab. So, what are some of the key uses of a Bunsen burner in microbiology? Sterilization. One of the main uses is sterilizing metal inoculating loops. When transferring microbial cultures, it's important to ensure that your tools are free of contaminants. A Bunsen burner provides the necessary heat to sterilize tools quickly and efficiently. Heating substances. Bunsen burners are also used for gently heating substances, such as liquids or media, in glassware. Whether you're preparing growth media or heating up liquid samples, the adjustable flame allows for precise control over the temperature. In conclusion, the Bunsen burner is an essential piece of equipment in microbiology labs, providing the necessary heat for sterilization and precise temperature control. It helps maintain a sterile environment, which is key to conducting accurate and safe experiments. Next up, we have the centrifuge. This powerful machine is used to separate different components of a sample by spinning it at high speeds, creating a centrifugal force that isolates particles of varying densities. From separating cells to isolating proteins and nucleic acids, the centrifuge plays a vital role in microbiology research and clinical diagnostics. There are several types of centrifuges, each suited for different separation tasks. Let's explore them. Benchtop centrifuge. The benchtop centrifuge is a compact, general-purpose machine commonly used in labs. It's perfect for everyday separation tasks, such as spinning bacterial cultures, separating plasma from blood samples, or isolating cells for various microbiological studies. Clinical centrifuge. A clinical centrifuge is designed specifically for handling medical or clinical samples, like blood or urine. It's mainly used for separating blood components, such as plasma and serum, or for preparing urine samples for further diagnostic analysis. High-speed centrifuge. If you need to separate smaller particles or work with larger volumes, a high-speed centrifuge is the go-to option. It spins at much higher speeds, making it ideal for separating subcellular components, like organelles, or for large-scale bacterial harvests. Ultracentrifuge. The ultracentrifuge takes it to the next level by operating at ultra-high speeds. It's used to isolate extremely small components, such as proteins, lipids, and even viruses. Ultracentrifuges are essential in molecular biology for tasks like purify viral particles and conducting detailed protein analysis. Some of the top brands for centrifuges include Beckman Coulter, Eppendorf, and Thermo Fisher Scientific. These brands are known for providing high-performance, reliable centrifuges for both clinical and research purposes. Centrifuges are indispensable in microbiology and clinical settings. Here are a few key uses of a centrifuge in microbiology labs. Separation of microbial cells. One of the most common uses is harvesting bacterial cells from liquid cultures. After centrifugation, the bacterial cells settle at the bottom, allowing researchers to collect them for downstream analysis, such as DNA extraction or protein analysis. Purification. Centrifuges are also essential for purify biological molecules like DNA, RNA, and proteins. By spinning samples at high speeds, scientists can isolate specific molecules of interest, which is crucial for genetic studies, diagnostics, and research. Sample preparation. In clinical labs, centrifuges are used for sample preparation. For example, they're used to separate blood components, like plasma or serum, for diagnostic tests, or to prepare urine samples for further analysis. In summary, the centrifuge is an essential tool in both microbiology research and clinical diagnostics. It helps researchers and clinicians separate, purify, and prepare samples for a variety of critical applications. Whether you're isolating cells or purify proteins, the centrifuge ensures the accuracy and efficiency of your experiments. Next, let's dive into one of the most powerful tools in microbiology, the PCR machine, also known as a thermal cycler. PCR, or polymerase chain reaction, is a technique used to amplify specific DNA or RNA sequences. This amplification process makes it possible to detect even the smallest quantities of microorganisms at the molecular level. There are several types of PCR machines, each with its unique features and uses. Let's take a closer look at them. Standard PCR thermal cycler. The standard PCR thermal cycler amplifies DNA through repeated cycles of heating and cooling, allowing the DNA to denature, anneal, and extend. This is the go-to machine for basic DNA amplification, especially for tasks like pathogen detection and genetic analysis. Real-time PCR qPCR. With real-time PCR or qPCR, you don't just amplify DNA. You can also measure the accumulation of DNA as it happens, providing quantitative data. This makes it ideal for tasks like quantifying microbial DNA, measuring gene expression levels, and detecting pathogens in clinical samples. Gradient PCR. The gradient PCR machine allows for varying temperatures across a sample plate, which helps optimize the PCR conditions. This is especially useful when you're trying to optimize conditions for new primers or DNA sequences, ensuring the best results.
High-throughput PCR. Finally, the high-throughput PCR is an automated, robotic version designed to handle large numbers of samples simultaneously. This is perfect for large-scale genetic testing and microbial identification, particularly in diagnostic labs. Stopwatch equals 2000. When it comes to reliable PCR machines, brands like Thermo Fisher Scientific, Biorad, and Eppendorf lead the way. These companies offer precise and user-friendly PCR systems that are trusted in both research and clinical settings. Now, let's explore the key uses of PCR machines in microbiology labs. DNA amplification. PCR machines are most commonly used for DNA amplification. By amplifying the genetic material of specific microorganisms, researchers can detect and identify pathogens even in low concentrations. Pathogen detection. PCR is particularly valuable for detecting microorganisms based on their unique genetic markers, such as bacterial species or viral strains. This allows for accurate pathogen identification, even when only trace amounts of DNA are present. Gene expression studies. PCR machines also play a key role in gene expression studies, where they can be used to measure how genes are expressed in microorganisms or host cells. This is essential for understanding how organisms respond to different environmental conditions or treatments. In summary, the PCR machine is a game-changer in molecular biology and microbiology, enabling the amplification and detection of DNA or RNA at a molecular level. Whether you're detecting pathogens, studying gene expression, or amplifying genetic material for research, PCR is an indispensable tool in the microbiology lab. Stopwatch equals 3000. Next, let's talk about a crucial piece of equipment in microbiology labs, biosafety cabinets, also known as laminar flow hoods. These enclosed workstations create a sterile, controlled environment, allowing researchers to safely handle potentially hazardous materials, such as infectious agents, without risking contamination. Not only do they protect the researcher, but they also safeguard the cultures and samples being worked on. There are three main types of biosafety cabinets, each designed to offer different levels of protection depending on the level of risk involved in the work. Class 1 Biosafety Cabinet. The Class 1 Biosafety Cabinet is designed to protect the user from harmful microorganisms. It allows airflow into the workspace to prevent exposure, but it doesn't offer protection for the sample itself. This type is suitable for handling low to moderate risk biological agents, like certain bacterial cultures. Class 2 Biosafety Cabinet. The Class 2 Biosafety Cabinet provides a higher level of protection, both for the user and the sample. It uses a laminar airflow to prevent contamination of cultures, making it ideal for handling pathogens in clinical microbiology labs. It's perfect for working with infectious agents, ensuring a sterile environment for both the researcher. Class 3 Biosafety Cabinet. For the highest level of containment, there's the Class 3 Biosafety Cabinet. This type has a sealed, glove box design that ensures complete containment of hazardous materials. It's used in labs working with highly dangerous pathogens like the Ebola virus or select agents, offering the most stringent protection for both the researcher and the sample. When it comes to reliable biosafety cabinets, brands like ESCO, Thermo Fisher Scientific, and LabConco are trusted in the industry. These companies offer a range of biosafety cabinets tailored to specific lab needs, ensuring maximum protection and reliability. So, what are some of the primary uses for biosafety cabinets in microbiology labs? Aseptic microbial culturing. Biosafety cabinets are essential for aseptic microbial culturing. They provide a contamination-free environment, allowing scientists to grow bacteria, fungi, and other microorganisms without the risk of external contamination. Handling infectious agents. They also protect lab workers when handling infectious agents, especially in clinical settings where exposure to harmful pathogens can pose serious health risks. Tissue culture work. Another key use is in tissue culture work, where mammalian cells or other sensitive cultures are grown. The sterile environment ensures that no unwanted microorganisms interfere with the growth or experimentation of these cultures. In conclusion, biosafety cabinets are indispensable in microbiology labs, providing both safety and a sterile environment for researchers and their cultures. Whether you're growing microbes, handling infectious agents, or working with delicate tissue cultures, these cabinets ensure that the work is done safely and contamination-free. Next up, we have an essential tool for microbiology labs, the pH meter. The pH meter is used to measure the acidity or alkalinity of a solution, which is crucial for a range of tasks in microbiology. Whether you're preparing media, testing samples, or ensuring optimal growth conditions for your microorganisms, monitoring pH is vital for successful experiments. Types of pH meters. There are different types of pH meters, each suited for various applications in the lab or field. Benchtop pH meter. The benchtop pH meter is the most common type found in labs. It provides accurate, precise measurements of pH, making it ideal for measuring the pH of liquid media, buffer solutions, or samples in microbiological experiments. Portable pH meter. If you're working outside the lab, a portable pH meter is a great option. This handheld, battery-operated device is perfect for fieldwork or testing small liquid samples, such as in environmental microbiology or water testing. Pen-style pH meter. The pen-style pH meter is a compact and easy-to-use device, resembling a pen. It's often used for quick pH checks on culture media or for field applications where simplicity and portability are key. Popular makes. When it comes to reliable pH meters, brands like Hanna Instruments, Thermo Fisher Scientific, and Oakton are well known for their high-quality, user-friendly devices, designed for both lab and field use. Uses of pH meters in microbiology. So, how are pH meters used in microbiology? Let's look at some key applications. Media preparation. When preparing media for microbial growth, it's essential to adjust the pH to create the ideal conditions for the specific bacteria or fungi you're studying. A pH meter helps ensure the pH is within the required range. Buffer solution testing. In microbiological experiments, buffer solutions help maintain a stable pH. A pH meter is used to test these solutions, ensuring that their pH stays within the necessary range for accurate results. Microbial growth monitoring. As microorganisms grow, their metabolic activity can alter the pH of the culture. By regularly monitoring and adjusting the pH of growing cultures, you can optimize conditions for growth and ensure your experiments run smoothly. In conclusion, a pH meter is a critical tool in microbiology labs, helping researchers measure and adjust the pH of solutions, culture media, and microbial cultures. Whether you're preparing media or testing field samples, a pH meter ensures that the right conditions are maintained for microbial growth. Vortex mixer, rapid and efficient mixing in microbiology. Now, let's look at a tool that's essential for ensuring thorough mixing in the lab, the vortex mixer. This device rapidly mixes small volumes of liquids, ensuring that samples in test tubes, vials, or other containers are well blended and homogeneous. It's especially useful in microbiology labs for creating uniform suspensions and reagents. Types of vortex mixers. There are several types of vortex mixers designed for different levels of mixing intensity and volume needs. Standard vortex mixer. The standard vortex mixer is the most commonly used. It's great for general-purpose mixing and comes with variable speed controls, allowing you to adjust the intensity of the vortexing. It's perfect for mixing small volumes of culture medium or bacterial suspensions, ensuring everything is thoroughly combined. Multi-tube vortex mixer. If you're working with multiple samples, the multi-tube vortex mixer can mix several tubes or vials at once. This is particularly useful in high-throughput labs where you need to mix large numbers of samples simultaneously, saving you time and effort. Vortex Genie. For more intense mixing, the Vortex Genie offers specialized vortexing. This model is designed for applications that require a more vigorous mix, such as when working with cell suspensions or during protein extractions. It provides more controlled and powerful vortexing. Popular makes. When it comes to reliable vortex mixers, brands like Thermo Fisher Scientific, Scientific Industries, Vortex Genie, and Cole-Parmer offer a range of models designed for precision, control, and durability in microbiology labs. Uses of vortex mixers in microbiology. So, what are some of the key applications for vortex mixers in microbiology? Microbial suspension. When working with bacterial or fungal suspensions, it's important to mix the sample thoroughly before plating or testing. A vortex mixer ensures that the sample is uniform and that cells are evenly distributed in the suspension. Reagent mixing. Vortex mixers are also commonly used for mixing reagents and chemicals. Whether you're preparing culture media, buffers, or other solutions, a vortex mixer guarantees that everything is properly mixed for accurate results. Cell lysis. For applications that involve protein extraction or DNA/RNA analysis, vortex mixers can help with cell lysis, or breaking open cells to release their contents. The vigorous mixing ensures that the cell membrane is effectively disrupted, allowing for the efficient extraction of cellular materials. Water bath, controlled heating for microbiological experiments. Let's dive into an essential tool used in microbiology labs for controlled heating, the water bath. A water bath provides an even and gentle heat source, allowing you to maintain a constant temperature for experiments that require precise thermal conditions. Types of water baths. There are a few different types of water baths, each designed to meet specific needs in the lab. General purpose water bath. The general purpose water bath is the most common and is designed to provide consistent heating, typically up to 100 degrees Celsius. It's perfect for general laboratory applications, like heating microbial culture media, reagents, or samples that need to be incubated at a set temperature. Shaking water bath. If you need both heat and gentle agitation, the shaking water bath is ideal. This type of water bath offers temperature control along with shaking, which is useful for experiments requiring both heating and mixing. It's commonly used in microbial growth studies, such as culturing liquid bacterial cultures where agitation is needed. Digital water bath. For more precise temperature control, the digital water bath comes equipped with digital settings for accuracy. This is perfect for sensitive experiments, like DNA melting or protein denaturation, where maintaining the exact temperature is crucial. Popular makes. Brands like Thermo Fisher Scientific, Scientific Industries, and Jalabo are well known for their reliable and high-precision water baths, offering options with gentle shaking and digital controls for more specialized applications in microbiology. Analytical balance, precision weighing in microbiology. Next up is a crucial piece of equipment for precise measurements in the microbiology lab, the analytical balance. This highly sensitive scale allows you to measure the mass of small samples with extreme accuracy, which is essential for everything from preparing media to analyzing microbial cultures. Types of analytical balances. There are several types of analytical balances, each designed for specific applications and levels of precision. Top-loading balance. The top-loading balance is the most common type of analytical balance. It provides quick and precise weight measurements for small samples. It's perfect for weighing cultures, chemicals, or media components in your experiments. Microbalance. For even greater precision, we have the microbalance, which measures ultra-small amounts of material with extreme accuracy, often down to the microgram level. This balance is ideal for weighing rare samples or tiny microbial cultures that require high precision. Semi-microbalance. The semi-microbalance falls in between, offering accuracy to the milligram range. It's often used for medium-sized samples or when working with sensitive chemicals in microbiological research that require high-precision weighing. Popular makes. Some of the most well-known brands in the industry include Mettler-Toledo, known for its high-precision balances, Bauhaus, recognized for accurate and durable balances, and Shimadzu, offering both analytical and microbalances for precision work. Uses of analytical balances in microbiology. An analytical balance is crucial for several key applications in microbiology. Media preparation. When preparing culture media, accurate measurement of chemicals and ingredients is essential. An analytical balance ensures you add the right amount of each component, maintaining consistency in your experiments. Microbial quantification. In microbial research, it's often necessary to weigh bacterial or fungal biomass for experiments. Using a highly precise balance allows you to work with exact amounts of cultures, ensuring the reliability of your results. Chemical analysis. The balance is also vital for chemical analysis in microbiological experiments. Whether you're weighing reagents for biochemical assays or other analytical tasks, having a precise measurement is critical for accurate results. Colony counter, quantifying microbial growth. Next up, we have the colony counter, a vital tool in any microbiology lab for counting bacterial or fungal colonies grown on agar plates. Accurate colony counting is essential for assessing microbial growth, determining colony-forming units, CFU, and quantifying microbial contamination in various samples. Types of colony counters. There are different types of colony counters, each suited to different lab needs and workloads. Manual colony counter. The manual colony counter is a simple but effective tool. It involves counting colonies by hand under a magnifying lens. While it's perfect for small-scale experiments, it can be time-consuming for larger plates with many colonies. Digital colony counter. A digital colony counter is a step up, offering a mechanical or electronic system that automatically counts colonies as you move the plate through the counter. It's ideal for high-throughput labs, improving both efficiency and accuracy. Automatic colony counter with software. For even more advanced counting, the automatic colony counter with software uses optical systems and specialized software to count colonies with high precision. It generates digital reports and is perfect for large-scale microbial assays or antimicrobial susceptibility testing. Popular makes. Some well-known brands that offer colony counters include Protocol, known for their automated systems, Fisher Scientific, which offers both manual and automatic options, and Stewart, which provides electronic counters with optical sensors for accurate colony counting. Uses of colony counters in microbiology. The colony counter is widely used for several key microbiological tasks. Microbial enumeration. By counting the colonies, we can estimate the number of viable microorganisms in a sample, which is crucial for understanding microbial populations in clinical, environmental, or food samples. Antibiotic susceptibility testing. Colony counting is also used to evaluate the effects of antimicrobial agents on bacterial growth. By counting the colonies after exposing them to antibiotics, we can assess how well those agents work in inhibiting bacterial growth. Microbial quantification. In various studies, colony counting helps to assess bacterial load in clinical samples, environmental samples, or even food products, which is important for safety and quality control. If you enjoy the content I'm creating, please consider subscribing to my channel to stay updated on new videos and content.